Hello, today we will be talking about phylogenies. So before we get into looking at the different groups of fishes, we have one kind of last major topic to look at. We've looked at systematics and taxonomy, we've looked at the history of ichthyology, and we keep talking about phylogenies and we'll be showing them, and it's showing you how different groups are related together. But we need to kind of review phylogenies a bit for those of you who are rusty on them, and for those of you who aren't familiar with them, to maybe kind of show you how to interpret them and the information that is located within them. So basically, phylogenies are going to show you a relationship between ancestors and their descendants. It's kind of a diagram of the genealogy of a group of organisms that you're looking at. So they kind of look like this, right? We've got this one line going up the side and then all of these branches that come off it. And the points along the one line that goes to the side, these are you can consider to be your ancestors. Okay, so this is kind of the more basal groups. And then from those you get descendants. So this green circle is a descendant from this red ancestor. And everybody above this line is related to this red ancestor in some way. This is the direct descendant coming off. Does that make sense? Kind of a way to interpret what it is we're looking at. We'll keep going through it in a little bit more detail. How do we infer the phylogeny? How do we know who are the ancestors and who are the descendants? Well, we have to do that by looking at characters. So we can look at the characters. Some of these characters will be analogous characters. So you can think of this as being like different ways that we see that help an organism swim. So in a shark and a penguin and a mammal, a dolphin, we see this kind of analogous structure for the flipper, right, that helps them. And we consider this to be homoplasy. We can have convergent evolution or parallel evolution. So convergent evolution is what we see in widely spaced lineages, the way that the shark and the dolphin have a similar body shape. The extinct ichthyosaur, which is an aquatic reptile, has that same kind of general body shape to it. They're not closely related lineages, but we kind of converged on the same shape that works well for this lifestyle. Parallel evolution, this is where we see similar traits in more closely linked lineages. This could be like in these two fishes, right? We see similar color patterns and similar morphology. They're two distinct species, right? They're found in two different lakes. They're more recently separated, so we consider that to be parallel evolution that we're seeing there. So these are analogous characters. We can also have homologous characters, and these actually have a common origin. So you can see this here shown in the limbs, right? You can see the bones, the orange and the yellow bones that are the same across there. So those have that common origin. We have other characters that we can use, and we've already talked about some of these with fishes, right? We've talked about those meristic characters. Those are the ones we can actually count. We see those change during development. We can count the number of rays, the fin rays in the dorsal fin, right? We can count the number of scales in the head. We can count the number of vertebrae. Morphometric characters, those are ones that we can measure. So these could be the snout length, it could be the standard length, or the fork length, right, to the fork of the tail from the nose of the fish. So there's lots of different ways that we can get measurements off there, right? We talked about body length measuring when we talked about mark recapture for age estimates. So there's lots of different characters we can look at when it comes to fishes. There are a whole suite of characters that we can use to help us determine what specific fish it is we're looking at. Remember when we worked our way through all those different anatomical structures of the fish? We talked about their bones, we talked about their swim bladders, we talked about their ear. All of these characters that we said you could use that to identify the fish. That's how we can identify the fish. Anatomical, osteology, soft anatomy, morphology. Some fish you can't tell in the field. You can hold it in your hand and you can say, I know the genus or I know the family. Some you can take right to species. Some you have to actually take the fish back and look at its vertebral count, count specific structures, measure things in order to determine what species you're looking at. In some cases, we're talking about molecular samples. You're going to actually have to run a DNA analysis on it. Some fish you can look at their behavior and get characters, ideas of which species it is you're actually looking at. We do see some obvious things. We talked about this when we were talking about the fins on the fishes, and you can see here the character state transformation series, abdominal pelvic fins here, well beyond those pectoral fins. We see the thoracic ones here where they're just about below it. And then we have the jugular where now those fins are in front. Yes, this is an identification character that you can use to determine the species of fish you're looking at. We can group some characters into synapomorphies and simplesiomorphies. So a synapomorphy is a character that's a derived character that's shared by a group. And this is helpful if you're trying to find or describe a monophyletic group, a group of tightly related organisms. Simplesiomorphies, those are a little harder. They're a shared primitive character. And the problem with primitive characters is sometimes descendants lose those. So it's hard to trace it into a discrete monophyletic group where you're trying to get the whole entire group from kind of the basal ancestral organism, including all of its ancestors. So simplesiomorphies can be a little more complicated because that primitive character may not be present in all of the descendants. The synapomorphies are definitely a little bit better because it's a derived character, so it is shared by all of the descendants. So synapomorphies you can use to build your phylogenies, those that possess that shared specialization, that's the synapomorphy, right? That derived character are united by having that character. So we can see here, right, if we have synapomorphy, the bill of a billfish, right, then everything else that's up above that is united because they have that. The alternative interpretation saying that you've got this one and this one and this one, but you don't kind of have this character at the start, it's considered to be not parsimonious. So when it comes to developing one of these phylogenies, you want to be parsimonious. That just basically means it's the fewest steps to get to that. So the thought is rather than, it's not like we evolved the bill 3 distinct times. The thought is the bill was evolved once, and then from that, we got all of our various billfishes from it, right? One evolutionary event that created all of these rather than 3 individual events. We do this because we believe this makes the most sense, that it would be the fewest steps, and that's generally how we determine which is probably the correct phylogeny, is that is most parsimonious. We have these 2 basic evolutionary processes that we see when we look at a phylogeny. We have cladogenesis, and this is your branching off from it, and then we have anagenesis, which is your differentiation along the branches. So cladogenesis is considered to be when you have that branching or splitting during speciation that leads to 2 or more lineages that gives you your sister species, the ones that are closely related together. Anagenesis is more of a gradual change that happens as we move along. So we have our differentiation along the branches versus our actual distinct branching. We do have to talk about systematic groupings. So if this is our phylogeny here and we've got our descendants and our ancestors, we use a bunch of terminology. I've been throwing out monophyletic a fair bit, and I'm hoping you understand that, but we're going to take a minute here just to make sure we're all on the same page. So if we're talking about a group that's holophyletic, this is a group that is the ancestor and all of its descendants. So that would be like this. This is our ancestor here, and it's everybody that comes from that descendant. Paraphyletic is when we're looking at an ancestor and some, but not all of its descendants. So that could be like this group here, right? So we have our ancestor, but we're looking at these individuals, but we're not looking at all of the descendants, just part of that group. Polyphyletic, that's when we don't have a common ancestor at all. That's when we're taking these guys here and these guys here, and we're going to talk about them, but they don't share one common ancestor in that group. So we have polyphyletic, paraphyletic, and holophyletic. So holophyletic and monophyletic are basically the same thing, where you're taking an ancestor and all of its descendants. The whole thing, or mono, just the one and everybody that goes with it. Okay, so holophyletic is kind of what we like. Polyphyletic we don't like so much. It's kind of hard when you have things that are that distinctly separated that you're trying to talk about as one group. Using those groupings we just talked about, when we're talking about supraspecific taxonomic classes, so any grouping that's above the level of species, should be based on evolutionary relatedness. That is, the group should be monophyletic. So these are our holophyletic and paraphyletic groups, not polyphyletic. So if we're talking about a group above species, so say we're talking about a family, right? Then we want a group that's going to include all the organisms descended from a single ancestor. So we want, like we talked about on the previous slide, our holophyletic, right? We want something that's paraphyletic. We've got our ancestor and we've got the organism, the species descended from that. We do not want it to be polyphyletic. This would not make a good group for a class or a family because you're missing all of these individuals in between that are most likely related to those on the end. Does that make sense? So polyphyletic is not a good way of grouping things, but anything that's monophyletic is. We have 5 schools of classification that we talk about. We have our traditional school, which is our kind of intuitive, our overall knowledge, the way things should be grouped together. We have phonetic, which is when we're looking at the overall similarity between the different animals. We have cladistics, so this is using our phylogenies, our cladograms, and how things are related together. And evolutionary. So this is our cladogenesis and anogenesis, that idea of how the different things branch off. And this is our quantitative evolutionary systematics. And that's pretty much what we do now, is we're using this cladistic method and the molecular method. And the problem is, it kind of is messing things up a little bit. We're getting some instability there. Your book talks a bit about phylocode, which is kind of an alternative way to organize the animals different from Linnaeus's method, and they say it would actually work better. The problem is we all like Linnaeus's method, and it's kind of hard to change at this point. An example of that instability that we see is if you just look at this, and don't worry about the names, this is just kind of to show an example. You see each of these branches over and generally branches into 2 groups. You get sister groups here, and that's what we're looking for. But this polytomy is the idea that this internal node would have more than 2, so it wouldn't just be sister groups. And we end up with, you can see here marked, this node that has a whole bunch of different groups in it, and it's trying to determine how those are actually related. And this is what the molecular analyses are giving us, is this kind of idea. And the thought is, this is not really very stable here. And they're recognized as orders, but some of that may have to change when it's resolved. And it may be a matter of looking at more data, more measurements, more molecular analyses. But we do like to put our things in groups. We like to have things in boxes and clear lines to determine which animal goes into which box. And the animals don't always agree with us on that. So as we work our way through the groups of fishes, you'll see that diversity, and you'll see how some things just can't really be labeled very easily. This was our overview of phylogeny. Hopefully now you have a better idea of how to read and interpret one of these phylogenies, and a little bit of understanding in what goes into constructing one. There's a bunch of new vocabulary in this lecture, so as always, if as you're reviewing it you have any questions, please let me know. Thank you.